to come up with the human genome. And today, and in the end, I basically introduced you to the ways in which, using first principle thinking, researchers were trying to speed it up. And I laid the foundation of second generation sequencing. So second generation sequencing, three basic sciences were used to speed it up. First way was to use mathematics, and it gave you the idea of the pigeonhole principle, whereby it suggests that if you dilute a sample enough number of times, you will be able to segregate single molecules inside a dilution. You can do this by compartmentalizing, micro compartmentalizing your sample. The other thing was chemistry. So most of the other earlier Sanger sequencing experiment worked on chemistry that worked on macromolecules or large amount of molecules. So you had to get, let us say, 50 nanograms of DNA in order to 10 nanograms of DNA in order to, for it to be um, observed on a gel. So now, if you want to work with smaller amounts, you want to work with some chemistry that allows you to do this. And here, different kinds of fluorescent chemistry was made, and a chemistry was invented where instead of looking at fluorescent signal, you could also look at, say, the electrical signal or the pH. And the third one, just like chemistry, physics was involved in order to use other alternative methods to detect and characterize individual nucleotide molecules, such as the current, um, because these nucleotides also act as conductive material and have a signature current flow that goes across these nucleotides when they are put under a voltage. So I'm going to talk about each one of these today. So the first one was enhancing efficiency by dilution. So here the idea was to dilute your samples, your library of DNA fragments, distribute or partition these fragments or this dilution into many small compartments, so that, and into small compartments, right? The compartments are small enough that you will get one fragment per compartment. You amplify the fragments within this compartment. So whatever amplification that you have to do, you use PCR often for this, and then you sequence using the appropriate method. So this entire process replaces cloning. This entire process also replaces the need to do individual Sanger sequencing, right? Because you are typically in a micro compartment. These compartments are microliter in size, so you can have thousands or even millions of such compartments. So how does it essentially work? It works by the process of, so they, in order to do this, people started to work with ways in which you could actually do PCR or isolate fragments in a small space. So one such process was emulsion PCR, whereby you could attach DNA molecules to a microbead. This microbead could be uh, diffused or captured in a water droplet that was immersed in oil, right? So basically, if you take water and oil, you will obtain these kind of small droplets. Now, in each one of these droplets, you would essentially have a bead and DNA attached to the bead, and you will do PCR. So now the same DNA fragment is going to have multiple copies within a droplet, and now you can do appropriate sequencing methods to see what's happening. So this is how it looks like. You can take each one of these beads, you can put them in picoliter wells. So I mean, again, remember, this is picoliters. So there is nanoliters, there is microliters, and then there is this picoliters, right? It's really small amount of volume. And then once you have a lot of copies of DNA, you can perform what's called sequencing by synthesis. So there are different ways of doing sequencing in second generation sequencing. The most popular one is called sequencing by synthesis. The idea is extremely simple. Whenever you have multiple copies of the same molecule in a small area, what you do is you convert double-stranded DNA after PCR to single-stranded DNA, and you use the single-stranded DNA as a template to perform a polymerization reaction, whereby the singles, you have bases added to this single-stranded DNA. So let us say this is your original library, which has been immobilized, as you can see here, right? So very often, most of these methods are called solid phase synthesis. Whenever a new base is added, let us say, so you have a primer, because you need to have a primer in order to do this polymerization reaction. And then whenever a new base is added, whenever you're synthesizing a new base, you have some method or some chemistry or physics based method which allows you to detect the addition of this base. Now, because whenever you will be sequencing, let us say you are sequencing this first well, because you have all the each one of these molecules is a clone of each other. Typically, the signal that you would be getting when each base is added to this single stranded DNA is going to be amplified by the number of copies that you have in that particular space. All right. So the way that people are able to detect the addition of a nucleotide in the process of sequencing or in the process of polymerization basically defines the different methods of sequencing which are out there, high throughput sequencing which are out there currently. All right. Does anybody have a question to understand or to which I can answer about this process? Do you see that there? Do you see some challenges happening here in this
Sir,
process?
like how this like you mentioned that signal whenever the base is added, there is a signal which is generated. So how that is generated? That is generated by the enzyme when it adds that base there, that base.
So I'm going to talk about exactly how it's generated, but I mean you or do all remember, right? It's a phosphodiester bond which is forming. So whenever a phosphodiester bond is forming, is there a particular group which is there a moiety which is released? So whenever you, for example, hydrogen and oxygen is combining, right, you get water, but that releases something, right? So for example, carbon and oxygen is combining, right? A hydrocarbon and oxygen is combining. So you often see that after combining hydrocarbon and oxygen, you have release of some water, right? As a byproduct. So
So in
whenever
that
DNA
case, also
polymerization is happening,
water.
can you, water is released? Something else
Yes,
could be
sir.
released as well?
The diphosphate.
Diphosphate is released, exactly. So, and diphosphate group is extremely reactive. So this is one of the ways in which you can control, you can actually detect, you can detect how phosphate is released. So that is one method that I'm going to talk about. But do you see some challenges here before I talk about these methods in this sequencing by synthesis? I can tell you, I can clearly go to the next step, but I want you to think about your engineers, right? So do you think it's just going to work like this? Can I just detect? So let
Detection
us
is
say we have these different wells, right? And in this wells, we are simultaneously doing this polymerization reaction. So how will I do this polymerization reaction? Any ideas? Would I add all the bases together in these wells and allow the
no, no.
polymerization to happen?
In one A, in one T, and G and C. In that way, we can do that.
So in each well, you will add each base. That's what you're saying?
Yes, sir.
How do you know which
Or
base to add when? You don't
or
know the sequence.
we can assign different colors to different bases.
Okay, you can
That
assign different colors to different bases. That is definitely one way. But how would you ensure that only one base gets added at a time? So
that is yeah.
each one of these sequences, each one of these clones of DNA fragments is different, right? So that means the sequence of each one of these DNA fragments is different. So one way, Ratnesh, that you were saying is that we add different bases to each of the microvels. But if imagine there are millions of microvels, so you will need millions of needles to do this, right?
So, sir, that can be done like that is the way it is done, right? Uh,
I'm not asking you what ways is, I'm asking you to think. So can you solve this problem? Okay, we don't want to use millions of needles. What else can you use? Let us say you have fluorescently labeled bases. How will you add bases to these millions of micro compartments? There are millions of these, right? So how will you ensure that only one base is getting added to each of these fragments? I'm asking you to think. Please think about it. This is very important. So one way is that the bases themselves are labeled, right? So the bases themselves are of different colors. The other way that I think Deepak, told that as a phosphate group is released. So we can detect the release of phosphate group, right? That's good. But how do you ensure that only one base is added at a time, right? Because you don't want to put a mixture of ATGC, right? Because if you put a mixture of ATGC, then the entire polymerization is going to happen. Are you guys are you folk understanding my question?
Yes, sir.
Everybody is understanding the question that I'm asking. How do you ensure?
Probably we
So I
could use
guess,
dideoxynucleotide for
yes, sir.
termination.
So like
Okay,
we
so
do in Sanger sequencing. So we can add some bases which have oxynucleotide and we can add some bases which are dideoxynucleotide.
but how do you decide which bases to add when?
No, sir, like we can add like all four bases, but like say half quantity is deoxynucleotide and half of it is dideoxynucleotide.
But then randomly, like this is not Sanger sequencing, right? In Sanger sequencing, if you have seen the video, that's why I wanted all you guys to see the video. In Sanger sequencing, you will get products in between, right? Some molecules are going to have
Yes, sir.
termination reaction. Some molecules are not going to have termination reaction, right?
Yes, sir.
But this
Yes.
is the difference here. In this sequencing by synthesis, you want to control exactly when a base is getting added. So the way that you guys are suggesting that we will do Sanger sequencing, where it's some kind of, okay, let us say there is one deoxy terminated base and if all the other bases are added, then basically what's going to happen is each molecule, let us say talk about this, right? One DNA molecule. Each molecule randomly is going to have different times at which you will see terminations, right? Just like a Sanger reaction.
Yes, sir. Yes,
So
sir.
how do you control? Can you think of a way? It's a very simple solution. It's one of those puzzle questions that you will be asked in an interview. How do you ensure that only one base is added at a time? Or you can detect one base at a time?
Sir, DNA's polymerase adds only one base at a time, and as it adds, so we can detect.
How will you detect? The DNA
Oh,
polymerase is adding, do you know the speed of DNA polymerase?
yes, sir.
It's adding 1000 bases in a second.
Yes, sir. So that
So
can be
we need to
detected,
have a
right?
method which is detecting the addition of DNA, like of DNA as fast as uh, yeah, as fast as polymerization. So that process you don't have. There is a much easier answer. So you have four bases, right?
Oh, I joined a little late, but maybe like we can just add the same base to all the microwells at the same time. So
Yes,
whichever
and
ones light up are the ones that have been added
exactly,
with the base.
exactly. That's it. It's that simple. So you are controlling what bases are being added, right? So in you can add one base and you can see which microwells are lighting up or which microwells you have a detection for that particular base. And you already know you are essentially having a camera. So most of these methods have high, like very sophisticated uh, microscopes attached, which are able to have a very high resolution, almost molecular resolution, whereby you can actually register, okay, this particular well had one color, this other well had a different, did not have a color. Okay, so for example, let us say we added T, right? And the different molecules out of these 6 molecules that I'm showing you, T was detected at this location at the first one and at the fifth one. So that would mean that at that particular cycle, you had a T in that particular molecule. Then you add, let's say you add a G, right? So a G maybe gets added in again in the first well, but it gets also added in the second well. So that means in that particular location at that particular cycle, you had a G in the pink one. In the red one, you had a T and a G, right? And in green, you had nothing. Here, orange, you had nothing. And in purple, you only had a T initially. And blue also, you neither had a T or a G at that particular location. All right. So these are the two simple ideas through which they were able to do this sequencing by synthesis. Idea that you are able to detect whenever a new nucleotide is added, and the idea that you are able to control what nucleotide is added when, because you are doing it yourself. So by this idea, one
So
of
I had
the
a question
first
here.
methods that came about was called
Excuse me, sir.
pyrosequencing. Yeah. Any question?
So, in the wells, if you add a base, so like that specific base stays in the well, right? So in the like next chain, it'll keep on getting added because it's already there
Very
in the
good.
well.
Good question. So what
So
one does is one adds the base, right? The polymerization happens, and typically this base is a chain terminating base, right? So basically only
yes, sir.
one base is going to be added at a time, right? Now what you do is now
TNT
you
base
wash
also.
off all the base you added. So that's why this process is happening in solid phase, because you can wash it off. Once you wash it off, then you add the chemical so that the chain extension can happen. Then you add the next base. Make
Sir, how
sense?
can
Okay,
we
okay,
wash?
sir, got it.
How can you wash?
Yes.
You wash. You basically wash with like water or some liquid, and
Okay,
everything
sir.
which is going to be washed away, which is free floating, you flush out essentially. So it's a
Okay,
chemical
sir.
reaction, right? So you have a aqueous solution with bases. You would remove that and you add a fresh solution which does not have those bases, so it gets washed out.
Yeah, sir. Sir, this process can even done even inside the cell, washing
It's
process.
inside the cell. So very good. You're jumping ahead. So the new sequencing technologies are actually doing that, but that is only been published like 1 or 2 years ago. If you're interested, I can talk more about it, but it's really that is really advanced, where people are trying to do sequencing within the cell.
Yes, sir. I have read like a paper on three-dimensional mapping. Oh, in that it was
You
mentioned.
have so much time, Ratnesh, to read papers relating to everything, but except for following the lectures in class. Good. We can
Okay,
talk
sir.
about it later on. Okay. Vishantan, you had a question as well.
So I already asked the question
You
that
asked the question.
how,
So understood, right?
yeah, how
So
do we
this is
move?
what
Yeah.
was the idea of pyrosequencing. So what happens is whenever you have a DNTP, you have iterative addition of DNTPs. So that is D-deoxy nucleotides. And when you add a deoxynucleotide to the DNA polymerase, you have a phosphate group which is released. This phosphate group was used to basically generate a light signal. So they basically you have a enzyme, a couple of a series of enzymes which basically interact with a protein called luciferase. And luciferase is called luciferase because lucifer, it's light. So it generates light. So whenever a base is added, a photon is released and that photon is detected with a nice microscope. So again, how does it work? You take your DNA, you shear or fragment it, you prepare a library. This library then has, because you are preparing a library, you are attaching, if you remember, the same concept as our first generation sequencing. You ligate adapters. So you ligate what's called short adapters to these fragments. These are called adapters because they allow you to attach to something. So these adapters are very often complementary to another DNA sequence or DNA probe, which is now immobilized on these beads. So now what you can do is you can hybridize these DNA libraries by heating them up and then incubating them with these beads. So what typically happens is you these libraries would then get attached to the beads, which are now you have encapsulated both the bead and the library inside an emulsion, right? Because of dilution, and then you perform the PCR inside this, and then you get these beads. Then what you do is you take these beads, you load them into wells, and you pack them with lots of enzyme, mostly DNA polymerase enzyme, and then you essentially add the bases. So whenever a base is added, that particular well in which you have the fragment corresponding to that base is going to light up. All right. So can you tell me what could be the limiting factor for the amount of data that is generated in this technique? So this was published in 2005, so 15 years ago.
So the speed of the process, because like you add, then you wash it off, then you add, then you wash it off.
Okay, speed of the process is a limitation. What else? I'm talking about the amount of the data in this case specifically. So speed is for the process itself, but what about the amount of the data? Can you see some physical limitations which are leading to some issues with the amount of data that is generated?
So take for example, we are sequencing 8 human genome. So there will be 6.4 billion, 3 billion base pairs. So for
No. This is nothing to do with the human genome. It's to do with the physical limitation of this platform itself.
So I guess the
Can you
number of wells and the are limited, and the beads itself can only accommodate like a limited amount of DNA.
Very good. So the number of wells is limited. So the number of simultaneous fragments that you can sequence depends on the number of wells that you can sequence at a time. All right. Very good. What else? So look at these beads. Look at the fragments which are attached on these beads. Can you think something about the length of these fragments which are attached? Can you have these fragments very long?
No, sir,
No,
because
sir.
we're using very small wells.
Right? You are using small wells. So if your fragments are long, they'll start to spill over in the other well, right?
but the wells are like 0.01 width and height, and everything will be there. So that is not a small for like DNA.
From where did you find the length of the wells?
Like you said that pico liter. So
That's
that
picoliter volume.
yeah, volume, but that can be
So
like
what does picoliter volume correspond to in linear dimensions?
0.0001 into 0.0001 meter. So that is around what is 0.1 in length into width into height.
So typically, so you can calculate that if you lay down DNA fragments
No, so
like
I
DNA
guess
molecules one after the
I
other
guess,
linearly,
so I guess
they
he took
are,
1 meter cube to be 1 liter, which isn't the case.
yeah.
Because 1 meter cube is
No,
1,000 liters.
yes, that is correct. But then that will be 0.01 into
So
0.01
think about it.
if I
Please
take
think. So
the
think
actual
about
liter.
it. So think about this thing, right? So length is also one issue because sometimes if you have longer molecules, they do spill in the other side. You can do the calculations. The other issue is that a lot of times the enzyme that you have added, right? Because this process is also driven by enzymes, these enzymes also might not last very long. So because these are actual biomolecules, right? So these biomolecules also have limitations. So the number of wells determines number of parallel fragments that you can sequence. The length of the fragment that you're attaching to the bead corresponds to the length of the fragment itself that can be sequenced. And the enzymes that you are also attaching also determine how long you can sequence. So if you have very long fragments, you need to have enzymes which last very long. Okay. So because of these limitations, this technique, which is called pyrosequencing, which was actually when I was doing my bachelor's. This is the technology that I first was using. They typically used to have much shorter fragments, so they would be having the sequencing would give you about a million fragments to be sequenced, and each fragment would be approximately 50 base pairs long, 36 to 50 base pairs long. So this was the first kinds of techniques, but it was able to generate good number of reads, right? So imagine a million reads, you're getting 36 base pair long. So that's 36 million base pairs that you are able to sequence. So that means if you have a bacterial genome, which is of size 1 million, how many times or what would be the coverage of sequencing that you would have done in this reaction, in this technique? So if you get 1 million fragments that you have sequenced, right? Each fragment is 36 base pairs long, right? And you are sequencing the genome, the DNA of a bacteria, which is 1 million base pairs in length. So what would be the coverage of sequencing here? So you remember we had done the human genome with 65x coverage over the period of 10 years that the human genome project lasted.
Yes, sir.
So in this
36 fold.
case, somebody answered.
36 fold, right? Because 1 million strands and each strand is 36 base pairs now.
Exactly. So it's 36x coverage, right? So you can clearly see how quickly with this idea of dilution, with this idea of parallelly sequencing many more fragments, you are able to enhance the amount of sequencing by millions of folds, right? And so fast. And this is only one reaction you have to do once you have to run the machine. So this basically gave us some very interesting, this gave us some new terminology and an interesting approach to sequencing, and therefore they called it what's called next generation or second generation sequencing. So the terminologies that I would introduce you to now here, the first terminology is called a read. A read is nothing but a sequence of genomic alphabet from a single DNA fragment after sequencing, followed by base calling. So base calling is the ability to or the process of converting your signal from, let us say, this kind of light phase or some analog phase into digital ATGC phase. This is called base calling. Okay. A read is what you get after you have done sequencing the DNA fragment. Read length is the length of the read. So when I say 36 base pairs, it is the length of the read. Advantages of this thing was that it was very fast, it was very accurate for that time, and it was great for small genomes, right? Small and simple genomes. So here again is a nice video. It's not a big video. So this pyro sequencing was bought out by a company called Roche and it was called 454 sequencing. Why did they do so? You can probably read about it. It's a very short video which will explain to you exactly how this thing worked out. And you can see this is how big that chip was, right? Which had about like lots of 100,000 or millions of wells. And this is how the sequencing looked like when you will be doing. So this color is what's called a pseudo color. So each well would actually give out the same light, right? But because you know that when I'm adding A, I'm expecting one color. When I'm adding T, I'm expecting another color. So they colored this image based on the different bases. And this is a color after adding in this is the color that you were able to they were able to obtain in one cycle. So whenever they're adding different bases, so right, this iterative addition, the when addition of all the bases finishes, that's called one cycle. Okay.
So what is the upper limit to like the amount of DNA that you can sequence using this in one cycle using one chip?
So upper limit of amount of so it's the number of wells that you would have on the chip.
No, sir, like exactly how much DNA can you sequence using one chip in one go?
How much DNA in terms of the amount of DNA?
So, yes, sir. So base pairs like.
Yeah, so that's what I'm saying, right? So a typical 454 reaction gives you about a million reads. Are you talking about a cycle? In a cycle, basically, million fragments are going to be sequenced simultaneously, right?
Yes, sir.
So basically, those million.
No, sir. Like basically, for example, you have 1 million wells, and each well has one bead, and say one bead has say 10 strands of DNA attached.
Okay.
So you can have 10 million strands of DNA that can be synthesized, and each strand is say, as you said, 36 base pairs.
Yes.
So you can have 3.6 billion base pairs sequenced
Yes,
in
but
one go. So
in
that
one
is
go.
the
But
way
the
I'm asking.
problem is that you want to amplify the signal of light, right? So if you are only detecting a single photon per well, then you need to have very good microscopy. So typically you
Right,
would
sir.
want to have in each bead, they have anywhere from 20 to 100 copies of DNA so that you get amplification of signal from that particular well. So parallelly, there are billions of molecules that are getting sequenced, but those billions of molecules, about 100, like there is 100x cloning of those molecules. So they actually belong to about a million,
Fine,
uh,
fine. Got it, sir. Got it.
million DNA fragments.
Got it, sir.
Yeah. So these were the advantages, but there were clear disadvantages. The reads were much shorter. they did not go beyond 400 base pairs. Typically, in a pyro sequencing, the reads was about 36 base pairs. It did not work very well for sequencing genomes, very large genomes, because what was the problem with large genomes? Can somebody tell me? What could be the problem with large genomes?
So we have to do multiple rounds of sequencing for that.
You have to do multiple rounds of sequencing, and also large genomes have a lot of repeats and such and stretches of DNA which
Yes.
are called homopolymer stretches, which was difficult for these machines to handle. Because if you have AAA, it will keep on adding A and it will not know what happened. Okay. Because at this point of time, I said that they were using chain terminating nucleotides, but I was incorrect. They were not using chain terminating nucleotides for at least first versions of pyro sequencing. So here, if there was like a lot of A's or a lot of T's or a lot of G's or C's, they would just keep on adding it in that DNA molecule. Okay, so then they came up with what's called Solexa sequencing. That initially it was called Solexa, and then it was being bought out by a company called Illumina, and now everybody knows it by the name of Illumina sequencing. All right, so they did something ingenious. So the
Solexa invented this, or they also bought it?
who?
Solexa.
Solexa invented it. Solexa was invented by an Indian and a British guy.
Yes.
I'm going to talk about the entire timeline of this. But
They
let me
got.
talk about the technology first. So in the case of pyro sequencing, the limitation was that you had to do this emulsion PCR, right? So you had to make this process very long. You had to make these emulsions, these droplets, right? Also, you need to have attachments of these fragments to this DNA beads, these beads, right? And so you need to have like, and these beads were typically much larger so that you could place them inside the wells easily, right? So, and this typically restricted the ability to the number of wells that you could, or number of individual molecular clones or DNA fragments you could sequence. So the Solexa team, they came about, they thought of a totally different solution. They said that, you know what, just let's get rid of the beads themselves and let's immobilize the DNA fragment itself on a surface, on a solid surface. Okay, so this is what they did. This is the basic idea of Solexa or Illumina. You have a DNA library as shown here. So you have two different molecules and you have a glass slide or a solid phase which has complementary short DNA probes attached to the adapters which you attach to the DNA fragments during the process of preparing your sequencing library, as you did earlier in Sanger sequencing, as you did earlier in pyro sequencing as well. Okay, now this particular adapter is complementary. So there are two different adapters, and this adapter is complementary to the uh, to this small DNA probes that are now immobilized on a glass slide or a solid surface. What you do now is there are billions, like hundreds of billions of this adapters on a glass slide. So this is how a glass slide would look like. Okay. And now what you do is you dilute your library or DNA fragments along with the adapters and you attach it on the glass slide. Okay. You just put it on the glass slide. So what would happen is just because it is so diluted, just by chance, two molecules are not going to be close to each other. Okay, these two molecules are going to have some social distancing between each other. Now what they do is, because they have adapters which are complementary, right? DNA probes which are complementary to these adapters, right? Which are put on the two ends of your DNA fragment. These DNA probes which are attached on the glass surface act like primers for PCR. Okay. Now, because this is a molecule, it's not usually like standing up like that, right? It's like floating around, it's wobbly. So because it's wobbly, and if you do a PCR reaction, given all the enzymes and nucleotides, what will happen is this one particular molecule is going to now bridge like that. It's going to form a bridge, molecular bridge. And then let us say this particular DNA probe, which is attached on the glass slide, is going to act like a primer, and now you have going to have a PCR product, a complete clone of the original molecule that you're going to have that was attached to begin with on the glass slide. And this, when this process happens couple of times, what you will be eventually getting is you will be getting cluster of molecules which are clones or exact copies of the original DNA fragment that was attached to the slide, right? So for example, there were two original DNA fragments which were socially distanced, right, on the slide. And now after a couple of these PCR rounds, this bridge PCR rounds, you are going to have a cluster of these clones, which will also be separated by a certain distance. Now you can imagine where I'm going from here. Each of these clones now, they can simultaneously now be sequenced, just like they were sequenced in the case of pyro sequencing or 454 sequencing. Just like that case, you are going to have, you can just sequence by synthesis now. But now, unlike pyro sequencing, in the case of pyro sequencing, you had wells, right? Right? So you knew where those wells were already placed on the chip that you had. But in this case, this distribution of molecules is absolutely random. You don't know where the molecule is going to go and bind. So what is the alternative now? So can you actually use pyro sequencing here? Can you use pyro sequencing in this method? Anybody wants
Perhaps
to
no.
answer? Okay, think about it. Think about if you can use pyro sequencing or not. You may be also able to use pyro sequencing if you can do some tricks. All right. So each
Sir?
dot here is it's called a cluster. It's called a cluster of molecules, right? Or a copy of a single DNA molecule that was bound to the glass slide that underwent what's called the bridge PCR. And this is the main one of the main ideas behind the Solexa sequencing or Illumina sequencing patent. OK, but before they went to all the way to this, there were certain key uh, key sort of reactions or key papers or key technologies that were developed. One of the key technology that was developed was the ability to capture a piece of DNA on a solid surface. OK, how did they do this? They essentially and you can see the lot of Indians involved in this process. So again, so this Indian guy here. So what they did was they had small DNA probes embedded in a agarose gel, right? And these probes are called capture probes. So they were essentially, let us say, complementary to a DNA sequence. Okay. DNA sequence, let's say they were complementary to DNA sequence B. Now, when you run a gel, right, because it is complementary to DNA sequence B, this DNA sequence is going to get captured in this area where you had these probes which were chemically mobilized, chemically modified by adding an acryltide group that binds the DNA probe to the gel, to the agarose gel, right? So that the DNA doesn't move away during the process of electrophoresis. So now what they showed was when they were incubating, when they had a target DNA molecule go through the gel, it is going to be bound in that particular layer. All right. So by showing this, they were able to demonstrate that yes, you can actually capture molecules of DNA in the solid phase in like a gel-like pattern, right? At a molecular level. So you can actually capture single molecules here, right? Because they were actually diluted according to they were diluted as much as single molecules will be able to capture them. So in a captured layer. So this innovation, the innovation of acryltide probes, which is what is used in even Illumina sequencing. So these probes which are attached, they are attached by the acryltide modification to the glass slide. This was one of the first ideas that demonstrated that you can actually put the probes on a glass slide, which can be then used to capture a complex mix of samples. So here they mixed two samples, A and B. B was having a target, A was not complementary to this probe, so they were able to only capture B here. Is this clear? Simple innovation. The next innovation that they
Yes,
were able to show
but
is
in this case, The probe can bind even if like one base pair is not matching. Like if the probe is like 20 base pair long, it can bind even there is if one base pair is not matching. So in that case, it will create errors.
It would there are yeah, so the amount of hybridization that you would require between the probe and the target, that is something that can be optimized, but that is not what I was trying to say here. So if there is an error, sometimes it binds, sometimes it doesn't bind. But in the case of sequencing, right, in the case of DNA sequencing, typically you would be adding adapters to each of your DNA sequencing library, right? So these are adapters at the ends of the DNA fragment. And these adapters are 100% complementary to the probe, which is used to capture the DNA library. All right. So the next.
So we already know that the sequence of that adapter.
Yes, yes, yes.
That's why we do.
You
Yes,
have
okay.
not read the previous class, no, Ratnesh? Go and revise the previous classes pipeline of how
Okay,
the Sanger sequencing was done, like first generation sequencing. Okay, so the next thing that they were able to do, and this was done by George Church, who is most likely going to get a Nobel Prize for this anytime. In his lab, a lot of this innovation happened there. So once they captured the DNA, right? So there is the variable region of the DNA, that is the library, and there are two constant regions as we showed, right? Then they captured the DNA in a gel like this at a single molecule level, right? And what they did was in the gel itself, they did the PCR reaction, right? So they essentially made what's called Polonese. So Polonese essentially is a DNA fragment which has been amplified so much that you can visualize it under a microscope, and it makes a small colony of cloned DNA fragments, right? With these variable DNA fragments. So again, these particular probes that was put on their acrylamide gel were acrylatide modifications, right, at the 5' end, right? And then these acrylatide modifications, they were essentially allowed you to immobilize your DNA library, right? So this is when they had zero amount of DNA library. Now they increased the amount of DNA library in the concentration, and you see that the more DNA library you add, the Polonese are more and more dense, right? So there is a ideal concentration of the amount of DNA library that you have to add in order to get well separated Polonese, right? So now that you had these well separated Polonese, the next innovation that happened was the ability to add and detect bases one at a time. And here they used what's called chain terminating. And this is where you added chain terminating dies. And here, this is the idea. So each die essentially has an OH blocked, and it has a fluorophore attached with a cleavage site. All right. So now and it's a phosphate, right? So whenever it gets incorporated in the DNA, you would have the fluorescence signal, but it is blocked just like the chain terminating reaction. But what you can do here is you do two things. First, you would be cleaving off the fluorescent signal, so that now once you have added the base, you don't need to have the fluorescent signal anymore. And you basically modify the specific blockage to make it an OH, so that the next base can be added, and the phosphate group, basically the phosphate group, can react to the OH end of the chain. So Illumina, basically this was the next innovation that was done, and they showed this innovation. Basically what would happen now is, whenever you will have a base added, so now instead of adding single base at a time like you did in pyro sequencing, in the case of Illumina, you had 4 different bases with 4 different colors, and in each cycle you will add all the entire mixture of bases, right? Now whichever base that will add is going to basically give that particular color. Once you have taken the photograph of that particular color, now you can cleave that particular color out. So now that base is not going to give you a color anymore. You're going to unblock the 3' end, the 3' OH, and then you are going to repeat the cycle again. All right. And as you can see, initially this was what the patent of Solexa was. So this is also called solid phase Sanger initially. Is this clear to everybody?
Sir? Yes, sir, it is clear. Sir, about you talked about George M. Church. So why he will get a Nobel Prize and not the inventor of alumina? Like, can
I'll
you
talk
please
about this later on. Okay,
go to the
Ratnesh?
last slide?
I'll talk about this later on. This is not related to the class. Is there something about
Okay, sir.
related to the class?
Okay, sir. Sir, like you showed that paper. That paper in was showing that in situ something like that. So is they are if they is they like they are doing inside the cell?
No, in situ means they are doing it. No, that is, it's not in situ. In situ essentially means that they are doing it in a specific region in a controlled manner. So in situ does not mean
Okay,
inside
sir.
the cell. That's called in vivo.
No, sir. In situ means cell inside the cell, but the cell is fixed, right?
But there is no cell here.
Okay, sir.
There is just a there is just a DNA
Yes.
lawn. There's a
Okay, sir.
gel and a lawn.
Okay, sir.
So besides this, any other question so far? Okay. So the next thing that they were able to show is by actually mixing this chemistry again in George Church's lab, the mixing this polyny, the ability to make polynies, and this chain terminating this particular solid phase chemistry. Now they were able to show that you can actually sequence the each polyny, right, based on the nucleotide that was particularly getting added at that in each polyny, right? Because each polyny is again exactly a set of clones of this same original molecule. And now what you can essentially do is you have these kind of 4-color imaging. So you image for the 4 different colors, right? Because this is fluorescent imaging, so you require very little, it's very high signal to noise ratio. So unlike in the case of pyrosequencing where you are just detecting a photon, which is common to all the nucleotides, here you are detecting a photon which is corresponding to the specific transition, fluorescence transition. So you need to have that the specific uh, emission spectra will be specific emission spectra will be observed for each nucleotide. So therefore you would do what's called 4-color imaging. You would combine and then you'll get an image. So for example, you have these 2 dots here, right? So in one cycle at this particular position, there was a red base, and in the next cycle at this particular location, there was a green base. All right. So now when you have something like this, that essentially means that you can now start to read what were the bases that were present at these locations. All right. Is this clear?
Yes, sir.
Yes, sir.
All right. So then came the seminal paper, which was behind Solexa. Solexa was already incorporated as a company. The paper usually comes after the patent. This is 2008 paper by Bentley and Shankar Balasubramaniam, the people who invented the sequencing technology and the Solexa company. So they essentially added this. So George Church and all these people, they showed that you can capture DNA and then you can do the sequencing of these polynies using 4-color cycling. And then Bentley and Balasubramaniam, they came up with the idea of actually doing bridge PCR. They perfected this chemistry. So instead of having modified acrylitide bases, they had some other modifications, and they were able to now very cleanly show that a molecule can come, it can sit on a glass slide attached to the probe, DNA probe. This DNA probe then can make bridges and can make copies, and then you can have like 2 different copies of this DNA probe, and then you can use PCR probes corresponding to the or like yeah PCR primers corresponding to the adapters that were attached to now begin sequencing. So for example, once if you want to sequence one fragment like this, right, you start to sequence, this is already complementary, so now a base would be added. You can now read this part of the read, right, or this part of the DNA fragment. Now this DNA fragment can then be hybridized on the other side, and you can have the complementary part to the adapter now like this, and then you can read the other side of the read like this. All right, so this is what's called the bridge PCR chemistry. So once you go back and you read this slides, you will be able to understand much more clearly. So this is the evolution of the Illumina sequencing today. So 1998, Solexa company was formed. Then you had the molecular clustering technologies were formed 2005. They sequenced the bacteriophage genome 5x174. If you remember, this is also the first genome that was sequenced. And nowadays, if you'll do the sequencing uh, analysis, very often this is still used as a legacy genome to check whether the machine is working properly. In 2006, they launched the first analyzer, and in 2007, Illumina, the company, acquired them. And you can see that after all this process is then when they actually published their technology for public consumption. So this is
Sir,
how
I
it
have a
looks like.
question.
What was the question, Ratnesh?
So the thing is that like in one paper I read that they have sequenced using Illumina, and after that they are checking that if Illumina is efficient or not using Sanger sequencing. So what's the purpose of that? Like Sanger sequencing is more efficient than Illumina?
So typically, whenever you are doing any kind of experiment, you would want to always check, right? If you are coming up with a new technology, you want to check it with the gold standard technology. So gold standard technology is a technology which is accepted by large number of people, right? So Illumina, Sanger was widely accepted by before Illumina came. Now, actually, uh, whenever you have a new sequencing technology that I will be talking about in the next class, let us say nanopore. So whatever the results of nanopore will be, people will ask you to check the results of nanopore by actually doing Illumina sequencing.
Yes, sir. Understood. Understood.
Yeah. All right. So just to repeat, each dot here actually on the glass slide, it represents a cluster of these molecules. Okay. After the bridge PCR, you have multiple copies of these clusters generated so that you have an amplification of signal, right? And then because you have the fluorescence based chemistry, you can add all the bases together, reducing the amount of time. And now because these clusters themselves are only as big as the molecule, the number of molecules you're putting, you can now go from, you are not restricted by the size of the well, right? But you are restricted by the size of the clusters that you want to obtain. So they increase the order of magnitude of sequencing from say 1 million molecules now to 3 orders of magnitude. Nowadays, you can actually get a billion molecules or a billion clusters getting sequenced at a time. So 3 orders of magnitude now it was increased, and going from a bacterial genome, now it's very easy to sequence a single human genome in a single day. All right. So this is how it basically works. You have your DNA fragments and you have these adapters, these DNA fragments. So your library of DNA fragments, you attach these adapters. These adapters are then you heat them up so that they become single stranded. The complementarity removes. Then there is a lawn glass slide with a lawn of complementary probes to these adapters. So when you incubate your single stranded DNA library on this lawn, again, you are basically these dense primers. So this is going to go and bind to this, the library fragment is going to go and bind to the lawn. And because it will be very diluted, so these molecules are going to bind far apart. Then you're going to basically add nucleotides, right? These are not fluorescently labeled nucleotides, so that you find a lot of clusters, right? There is bridge amplification and you have a lot of clusters formed around the same molecule. And the assumption is because initially the molecules were not close to each other, these clusters would be basically having the same molecule, right? So you will have the signal amplification. Now after that, you start to add your fluorescent chemistry, this chain terminating chemistry. And now let us say you have these 4 clusters, as you can see. And now depending upon what base pair, right, or what nucleotide was getting sequenced at that time, a corresponding color is going to come up, right? So let us say there are 4 clusters here. In this top cluster, a T was added, so you will see a green color. In this bottom cluster, a G was added, so you see a yellow color. In this cluster, there was a C added, so there was a C, a blue color, and A corresponding to red color, right? So now you do this, you take a photograph. So this is the photograph that you get. Then you cleave away the fluorophore, right? So you cleave away the fluorescence giving molecule so that now the cluster is not going to have any color, and you remove the base blocking. Now you add the next base and you take another photo and you keep on repeating it, right? So now if you specifically look at this particular cluster, the sequence that you will be getting is G C, T, G, and A. Okay. So this is how you would know that this particular cluster actually had G, C, T, G, A, right? So on and so forth. That particular fragment attached there. And like this, you will have billions of fragments getting sequenced simultaneously. And the process by which you are able to now say, okay, that a G is added here, this is called base calling, because you're going from the image space of a yellow color to actually assigning a base, that is G to it.
So we have another class now.
Just give me a second. I'm sorry for the bell. Yeah. Please ask the question.
So we have another class now.
Yeah, sorry. So yeah, I'll end here and we can take it up from next class onwards. If there are any questions, you can stay on and I can answer the questions. Otherwise, yeah, please go on. Thank you.
Thank